Awesome. I think we're recording.
Yep.
All right. Well, thank you, Joe, for that great introduction and also for the invitation to come speak today. Yeah, as you hinted at, I'll be talking about some of the millisecond informatics pipelines that we're working on, so including real-time search. Then really what I'm going to talk to you about is like essentially how we can apply all that. So in action, basically referring to what are all the applications now we have for mapping dynamic proteomes, particularly using sample multiplexed quantitative proteomics. Now, the key to this, right, is the ever-present challenge in one dimension of the huge kind of coverage of protein abundances, right? So 8 or 9 orders of magnitude covering the proteins as opposed to 3 to 4 for mRNA, which is inherently a challenge, but then coupled on top of that with the fact that we have dozens or hundreds of post-translational modifications or splicing events that can generate millions of proteoforms, leads to a landscape of diversity and abundance that we need to track and be aware of in developing quantitative proteomics methods. Now we can essentially tile this into a honeycomb of factors, right, that govern this landscape. So in particular, I mentioned, right, protein abundance. This also plays then into protein degradation, and both essentially affect our measured amounts of protein. We also then have factors in terms of post-translational modifications, right? So FOS and ubiquitin and oxidation. But on top of this is layered a host of other factors, including protein kinase activity or protein enzymatic activity for proteases and ligases, as well as subcellular localization. So different proteins in different spaces serve different functions. Then we also, of course, have with the hints at protein cross-linking and cross-linking mass spectrometry, trying to study binding interfaces and conformational changes within these proteins. And last but certainly not least is trying to interpret essentially protein-protein and protein ligand interactions. And all of this essentially encompasses the many factors in proteomics that we'd like to essentially tackle, but not everything is kind of ready for the limelight, right? So the canonical proteomics pipeline just classically looks like this. We have changes in cell state with, for example, activation of these T cells on the top, or post-translational modifications that we'd like to measure. And in measuring, also try and understand the landscape of what these proteins are potentially doing in a given sample. Right, so we isolate the proteins, digest, do LC-MS, MS. But for the most part, many of these pipelines, right, are agnostic to the input samples. So you run the same label-free analyses, whether or not the peptides you're putting in are yeast or human or mouse or come from bacterial species. So one of the main questions that we asked was, can we use biological information in our MS pipelines, right, to improve kind of the landscape of what we understand? And in part, quantitation helps us do this, right? So we can get spatial and temporal and conditional effects read out in terms of protein quantitation, right? And so just a background, most everyone's familiar by now, but just to ensure those who aren't, right? So we're going to talk about sample multiplexing for quantitation. So TMT Pro, the latest round of TMT reagents from Thermo allow for multiplexing of up to 18 different samples simultaneously. I'm only showing 16 because this slide is more than 3 months old. But essentially we can take now 16 or 18 different samples, quantify each one of these in terms of their relative abundance. And what's particularly nice about this, right, is like our high accuracy quantitative measurements can be accomplished across multiple conditions or across time series with replicate analyses inherent to them. So this brings along with it 3 kind of classic advantages with TMT, and that includes throughput. So one analytical run generates quantitative information for now up to 18 different samples, so inherently higher throughput. We have fewer missing values because we get quantitative information for every single sample when we run these analyses. And last but not least, we can do just what I hinted at, which is use or implement complex experimental designs. So we can go after multiple doses and treatments and go after mutants, et cetera, all in the context of a single experiment or a single plex. Right, and so that helps us in part begin to solve kind of the spatial effects, but again, that's kind of post hoc. So it doesn't help us solve the limitation that the instrumentation when we were addressing all these problems, could not learn or interpret biological information. And then if it can't learn or interpret that, it can't use that during real-time analyses. And so one of the cores to this for us was essentially the fact that we have post-hoc assignment of biological information, right? So we generate spectra, right? And then we match and score using whatever your favorite database is, so Comet or Sequest or Andromeda. And then we do all our filtering post-hoc from that to find the highest scoring peptides and the ones that have the lowest FDR from the sample. And in the end, then we generate afterward, right? After our acquisition has expired, after our samples have been collected, we now generate our peptide information, which is relating spectral information to actual biological information. But of course, because this all happens afterward, there's no actual integration of the biological features into the mass spec acquisition. And that's really the goal and fundamental to kind of what I was doing in my work with Steve when I left, and then what my work or my group is now doing. And so we can imagine this also just in terms of building smarter instruments is essentially just coupling, right? So we have raw data that the instrument is generating, and all it sees is essentially 2D matrices of M over Z and intensity. And we have matched peptides and all the in silico work we can do in a data system post hoc. And we just want to use intelligent data acquisition or millisecond informatics to couple these two sets of analysis, right? So we want to be able to couple our raw data to our matched peptides, but we want to be able to do that in real time with our acquisition so that our acquisition becomes smarter and our post hoc data analysis can use that smarter, faster, more rich data moving forward. Right, and so essentially, anytime we're trying to recreate a system, we'd like to recreate the entirety of that system. So offline for our data systems, when we're essentially matching spectra to peptide, what we're trying to do is a raw data conversion, essentially estimate to the best of our ability the monoisotopic peak, match that monoisotopic peak and its fragment spectra to theoretical spectra, and we're using Comet to do this, and then false discovery rate estimation and quantitation. And so our real-time search capabilities had the goal of doing all these exact same things with all the exact same protocols, right? So we want a one-to-one match of what we're doing offline to match what we're doing online. And so the first and largest step of this was adapting a current, really well-known search algorithm, so Comet, which was based on and similar in many ways to the original Sequest that Jimmy Yang had developed. And in collaboration with Jimmy, we essentially adapted Comet to allow for real-time search, or allow for speeds that could be used in real time with data acquisition. So we're talking about milliseconds per spectral search. And we'd like to do matching and scoring and filtering all online. And we showed that we were actually able to do this, right? So last year we published all this work, essentially showing that we could get single search or single scan search speeds on the order of about 7 milliseconds for yeast and about 15 milliseconds for human. And this was kind of the first shot at this. And so ever since we've gotten better, our databases have gotten more efficient in terms of our ability to search them. And so these are now down to the low millisecond, basically lower than C# can natively measure in terms of search speeds, which means that we can search larger and larger databases as we move forward. And so now, of course, we don't have to wait to search spectra. We can essentially use this information because we're acquiring it and learning it on the fly to actually inform our acquisition. And so that's essentially what this platform we built, termed Orbiter, did, right? So it used all this spectral information to generate biological information in the form of peptide sequences, and then used information about these peptide sequences to inform whether or not we should collect further scans. And so we showed that in the context of multiplex quantitative proteomics, we could get essentially a doubling of instrument efficiency, right? In that if we take a normal SPS-MS3 run, right, so high accuracy quantitative run, and we cut the gradient time in half, so from 180 minutes to 90 minutes, and then use Orbiter or real-time search with those shorter 90-minute runs, we could still quantify about the same number of total proteins, so over 8,000 quantified total proteins at the end of this, but we could do it in half as much time. And so that effectively means our instruments are twice as efficient. So you gain back that time to now explore another plex. right? So we also showed then that you get really consistent quantitation versus the SPS MS3, which is kind of the standard for accurate multiplex quantitation. So we actually found that in a few examples, Orbiter, because we're able to do kind of smart fragment ion selection for our SPS ions, could improve quantitative performance. And so we show this here for endocrine and alk2, just two proteins that were outliers for our SPS MS3, where they didn't show a very large change, but they show a dramatic change when we use the more accurate real-time search. And so that's kind of the basis of what I'll talk about and some vignettes coming up. But the idea is essentially now we have a millisecond informatics platform that does everything you can imagine online. So in the span of 1 to 10 milliseconds that we were doing offline on a server or instrument computer. And so, of course, Thermo has leveraged this, and now it's available in the Orbitrap Eclipse for general use for real-time search, and we're excited to kind of see it out there, see it being used. So now that we can essentially integrate these biological features, and now that we can do this kind of one-to-one online-offline match, it was how can we essentially begin to apply these systems to interrogate proteomes, right? And so we have these diverse proteomes we'd like to be able to use faster, more efficient searching, but also intelligent searching to take advantage of that to dig into these proteomes. And so the current state of it, right? So we have this diverse and dynamic body of biomolecules, and we can use real-time search, but how can we essentially use it to detect causal agents of cell state? And so to get at this, I'm going to talk to you about essentially 3 vignettes. These are partially collaborations, partially work from collaborations in Steve's lab as well, and then some new work we're doing right now. And we'll start with explorations of the aging proteome, and particularly explorations of the murine or mouse aging proteome. Right, and so the big question we were asking with this study is, how does age affect proteins and protein complexes over time? So what happens to proteins as we or other mammals age? And it's hard to do these studies right on a person because, one, you can't do biopsies necessarily on all the tissues you want, but 2, tracking for that long would be very, very expensive. So of course, instead we chose mice as our model system for this. And we didn't want to just choose one tissue from these mice. So looking at 2 different ages to start out with, so about 34 weeks and 79 weeks, right? We didn't want to start with just one tissue, and we didn't want to start with just a pair. So looking at the brain and a few sections within the brain, we wanted to start with something that encompasses most of the biology or the fundamental organs in mouse systems. So what we did is we built essentially a panel of about 200 samples covering 10 different tissues, right? And across essentially 20 mice. And so these 20 mice are across ages as well as both male and female mice. And so then using real-time search, what we were able to do is search and analyze all of these, get them through the mass spec, so all 200 samples across our system in about 2.5 weeks, with something around 1 proteome every 2 hours, which is a pretty good rate overall, especially for tissue proteomics. And so just showing you kind of an overview of the kind of descriptive stats of what we found, right? So nearly 350 LCMS runs, 9 million spectra, and just about 12,000 quantified proteins that we were able to identify. And I should say, very importantly, this is a collaboration between my lab and Steve's lab and Gary Churchill's lab and Ron Carstangi's lab at Jackson Labs, with some diligent work by Greg Keel, who's leading the bioinformatics effort now. So here's just a picture too of essentially the total quantified proteins across our different tissues. So you can see we got pretty good coverage across these tissues for all of our mice. And importantly, what we can begin to do once we have such a large and rich data set is to decompose this into some of the underlying biological features, right? So the most obvious is, of course, the effects of sex on proteomes across these mice. And so we can very easily see, just doing some principal component analyses, that we can cluster very nicely male versus female mice, something that's totally expected and relatively easy, but good to confirm. We can see that we see a large number of proteins that essentially differentiate male and female mice. And so I should say this is also just an example in one tissue, right? So this is just the kidney. So we can go on and look even further, right, at this PCA analysis. And if we dig a little deeper, so now if we look into PCA4 or PC4 in kidneys, we can see that we can also pretty well cluster our two groups, right? So young and old mice away from each other. And we can see, in fact, that the changes in the proteome here on the right side for these, right? So the kidney example, we can see hundreds of proteins that essentially differentiate old and young mice across these systems. And so that was really encouraging for us, right? So if we can differentiate proteomes in this way, that allows us to begin to understand not only something that's really well studied, which is essentially sex as a biological variable in mice, but also how age plays a role as a biological variable in these mice. And you can see just highlighting a couple proteins in there. And so more importantly, what we can see is because we have essentially such a rich tissue subset, right? So covering all 10 of these tissues, we're able to look and try and find clades or proteins that seem to show either pan tissue responses. So you can see one of those where we see an immune response that's generally upregulated in many of these tissues here in this section. But we can also look for tissue specific changes. So you can see here in the fat section that there's actually a clade of proteins that seem to be specifically responding by being downregulated or their abundance being reduced as a function of age. And so we can actually see that many of these proteins too, as one might suspect, are involved in metabolic processes, specifically oxphos and general components of the 5 complexes in the oxphos system. And so what this was, again, really encouraging of is the fact that tissue-specific age effects can be profiled in this manner, which suggests that we can begin to tease apart exactly kind of what this age as a biological variable looks like across each one of these tissues individually, but then also trying to understand more globally how age is affecting the biological and organ systems in these mammals. And so, right, we can pull out specific proteins and look for changes that seem to be happening across all these tissues. So again, these pan tissue responses. So we can see that immunoglobulins, at least IgKC, is upregulated in essentially every tissue that we monitored. Not just seemingly for everything we've done, not just a function of increased blood or anything like that once we control for that. So this is a real response of essentially what looks like increased immune proteins in these tissues. And these consistent age responses were interesting, but another interesting aspect, right, is inconsistent age responses. So if we look then at this other protein that we highlighted earlier in the volcano plots, this BCAT1, so it's a branched chain amino acid protein, we can see that in all 3 brain tissues, right, we see essentially no change across the 2 ages, 2 age groups. But if we look in kidney, we see a significant decrease in protein abundance as mice age. And we see an inversion of this, right? So in fat, we see a significant increase in protein abundance with age. And so these tissue-specific changes are also particularly interesting because any age effect that is tissue-specific is something that we love to understand further, particularly as it relates to, in this case, right? So BCAT1 is a branched chain amino acid protein that has previously actually been found to play a role in aging. And so previous work has shown just generally when they looked at mice, they saw a decrease of mRNA expression of BCAT1. And if they knock it out, they actually see in worms that the worms live longer. So it definitely seems to be age or tied to age and has an effect actually if you knock it out on aging. But it allows us to begin to ask open questions, right? So if we see differential effects in kidney and in other tissues, how is this coming about? So aging is driving a change in protein levels, and we can now ask, where is that coming from? So is it coming from a reduction in the number of mitochondria? Is it affecting ATP levels and other potential metabolic signals or pathways. And so last but not least, what's particularly interesting, right, is so the age-specific decrease in BCAT1 in the kidney is really interesting and consistent with previous work, but this notable and significant increase in BCAT1 protein abundance in adipose tissue or fat strikes us as something worth kind of following up, just because, all right, it's well established that BCAT1 goes one way, right? It goes down, but what does that mean in fat that this is all of a sudden going up? And teasing apart those consequences is something we're hoping to follow up on in the future. Now, next, we can again pull out specific examples of other proteins that seem to be affected. So EMC1, so the EMC is the ER membrane complex, which is an insertase complex and is responsible in part for protein quality control, particularly for transmembrane proteins. And we can see that in the cerebellum of these mice, EMC1 abundance increases, right, compared to the younger mice. And so what's striking about this then is that we have a protein responsible for quality control that seems to be upregulated, or its abundance increased as a function of age. And so we can now begin to ask other questions, such as what is aging doing? So it increases EMC1. Is it essentially leading to more stabilization of transmembrane proteins? And so why this is particularly interesting is EMC1 has already been tied to issues of cerebellar atrophy, which suggests that maybe modification or engagement of EMC1, or particularly maybe even stabilization of this, could aid aging and reduce kind of neurodegenerative issues. Now, not only did we want to take a look at kind of these protein-specific responses, but we were also particularly interested in trying to look at kind of a larger picture of how these proteins might be interacting. And so what we wanted to do is generate a complex centric view of murine aging. And to do this, what we wanted to do is look at essentially co-abundance across our mice as a response to aging. And so if we can look at essentially the correlation of proteins within a given complex as a function of aging, we can begin to understand how the stoichiometry of these proteins might be affected over the course of an animal's life. So essentially, then how does aging affect these protein complexes? And so here, then, it's just one example, right, of CCT, or the chaperonin-containing T complex. And so on the left, we see the correlation map across each one of the young mice, right, for proteins within the CCT complex. And you can pretty easily see that there's essentially two general subcomplexes in here, right? So CCT4, 7, 8, TCP1, and CCT3 are one, and then CCT6A, 2, and 5 are the other. And so in young mice, these seem to be pretty well differentiated. But as mice age, we see a higher correlation of these two subcomplexes with each other, suggesting potentially, right, that these two subcomplexes are coming together. They're binding more readily, and the stoichiometry of the entire holocomplex is upregulated. And we can begin again, right, to tease this apart. So not just for CCT, but look at all protein complexes, say in quorum, and begin to try and understand how these protein changes are affected by aging, right? So not just protein abundance, but also protein stoichiometry within complexes. And so once we begin to establish this, we can take larger and larger views. So here's all of the tested protein complexes that we looked at, and essentially just this estimated age effect on stoichiometry, so the relative difference in correlation. You can see very nicely that the CCT complex shows a large change in correlation overall, but we see a host of other proteins in here that we can now kind of dive into. And so the idea is to release these type of work as a resource to allow kind of the murine aging community to explore and investigate this further. All right, so the second little vignette that we're going to explore is essentially looking at how we're engaging with thermal proteome profiling to look for protein ligand interactions. Now, of course, thermal stability is nothing new. It's been around for for decades in terms of single protein analyses, but of course, Savitsky and Banchev popularized it through the development of CETSA and TPP. And with the expansion of the TMT pro sets, we really wanted to take advantage of essentially an extension of TPP, which Roman Zubarev's group had developed, which is the integral stability assay or protein integral stability assay, right? And so instead of building all these sigmoidal curves as a response to temperature, what the Zubarev lab suggested was essentially integrating under the curves, but doing this methodologically to build up essentially single abundance profiles as a response to temperature, right? And so this area under the curve, or this methodological area under the curve, could then be a readout for protein stabilization or destabilization. And what was super appealing about this, right, is, of course, if we're going to push all 8 or 10 of these channels down to just a single channel by combining and mixing each one of these temperature points, that means that the more essentially channels we have, the more conditions we can explore. So we can explore full dose curves or look at how essentially stabilization is really affecting a lot of different systems. And so Xiaoming Li and Jonathan Van Brinken in Steve's lab showed this really nicely in the TMT Pro paper, right, where they were looking at essentially response to kinase inhibitors. So treat with staurosporin, in this case a HCT116 lysate, and try to observe what proteins are stabilized or destabilized as a function of staurosporin treatment. And so they used the PISA assay and looked for essentially consensus stabilization. And now we're able to do it with a control and up to 15 different treatments. But in this case, just replicate analyses of 2 treatments. They were nicely able to show that essentially you could get improved coverage. So right, the order of nearly 7,000 total proteins with stabilization information, right? And you could particularly dive a little bit deeper into this and assess, in this case, right, staurosporin. So we know we're looking for kinases as an ATP competitive inhibitor. So now looking at just all the quantified proteins, or all the, not just the quantified proteins, but the quantified kinases, I could see specific stabilization and destabilization of a subset of kinases at both the 1 micromolar staurosporin treatment and the 20 micromolar staurosporin treatment. And begin to tease out that, say, the effects of CAM kinase 1 stabilization requires a full 20 micromolar of staurosporin. Now, having seen this and worked with them on using real-time search for those PISA assays, what I was interested in is when I got to the University of Washington, Cole Trapnell's group had recently published this idea of Cyplex, which is single-cell RNA-seq, but on the analysis of large or relatively large libraries of chemical treatment of diverse cell lines. Right. And so the idea is essentially take, in this case, 3 cell lines and 188 compounds across a number of doses in replicates, and then analyze single cells for their RNA profiles. And so just one I'm going to highlight, because we'll come back to it, is epigenetic regulation, which has the largest cohort of inhibitors in this study. But what they were able to see is that, again, doing PCA or principal component analysis, they could pretty well differentiate and cluster chemical treatments based on this PCA analysis, right? So the green cluster corresponds with an HDAC inhibitor, varinostat or SAHA. The blue is dexamethasone, and the red seems to overlay with Nutlin 3A. And so seeing this and seeing the kind of diverse response of these cells, right, and how many of these overlap. So Nutlin and BMS seem to nearly completely overlap, but there's still some green dots in the Nutlin kind of cluster at the top. What I was really curious about, and what my group has begun to collaborate with Jose McFarlane Figueroa, is the idea that even within these, there seems to be a large degree of heterogeneity, right? So there's a relatively diverse response of these cells to small molecules. So an open question for us is, how can we further understand why there is this large heterogeneous response of cells to specific small molecules. And how can we use proteomics to further understand that? And so the idea is for a subset of these, in collaboration with Jose and Cole's lab, to take some of these, and in this case, again, right, the epigenetic regulators, so HDAC inhibitors, and use those to try and understand protein ligand interactions, in this case, protein HDAC1 interactions. Right, and so HDAC inhibitors are essentially just going to inhibit decetylation, which is going to stop compaction of DNA or chromatin. And so we started off with these experiments. So Rose Fields in my group looked for stabilization as a function of HDAC inhibition using protein integral stability assays, and was able to very nicely show that a small cohort of proteins are significantly stabilized as a function of HDAC inhibition, right? And so we can see particularly HDAC 1, 2, and 6 are all stabilized as a function of HDAC inhibitor binding. But we also see a cohort of other proteins that are notably stabilized and destabilized as a function of HDAC-I treatment. And so further investigation of this can help us begin to unravel those heterogeneous responses from other large-scale data sets. And inherently then the integration of these two might further explain how or what exactly the cell state disparities are in these systems. So we could nicely correlate to these results, right, with previous data, in this case, vorinostat, looking at the fact that KIs for vorinostat were lowest for HDAC 1, 2, and again 6, and then 3 actually shows a differential response. So that's worth kind of following up with to see why that is, and why that wasn't further stabilized. All right, so the last vignette I'm going to get into today is essentially some work where we applied real-time search to profile reactive cysteines in systems, and this was recently published earlier this year in Nature Biotech. And so essentially the profiling of cysteine reactivity was started by Millisecond based on developing a new chemical probe to improve the activity-based protein profiling assays that Ben Krabat had significantly developed over the last few years, right? And so essentially Millisecond built a platform for streamlining this, which he termed SLC ABPP, for streamlined activity-based protein profiling. And essentially what it does is it combines the utilization of fragment libraries, right? And also TMT labeling. In this case, again, TMT Pro now allows for 16-plex or 18-plex versions of this. And in a control sample, essentially what happens is DMSO or other vehicle is used, added to the control cells, or control proteins. These proteins are then treated with an iodoacetamide-based probe. So it's iodoacetamide with desthiobiotine, or DBIA. This probe then labels all available free cysteines. And then when you go to analyze it, that should be the maximum, essentially, abundance of labeled cysteines available, right? So if it labels everything free. In the treatment cases, what you can do is add electrophiles, specifically electrophiles for cysteines, so in this case, acrylamides and chlorocetamides. Treat those samples with a library of these electrophiles, and then again add the DBIA or iodoacetamide-based probe. And essentially, anywhere that those electrophiles, these library electrophiles have bound, will take up one of these thiol residues and lead to a reduction in the abundance of a given peptide. And in this way, you can then label with TMT and mix and do a single avidin enrichment of the pooled samples, and then analyze that in a single shot with real-time search, right? So real-time search allows us to increase the throughput again by specifically targeting peptides that we know to have this DBIA tag. And so what Millisecond was nicely able to show is as soon as you kind of work this pipeline and reduce essentially the acquisition time by reducing essentially the throughput, or increasing the throughput and reducing the amount of time you need for mass spec, you can begin to analyze nearly 300 different compounds in just about 3 days, right? So compared to original ABPP, that's about a 42x improvement in the number of fragment based fragments that we were able to profile in a given amount of time. So incredible improvement in terms of library throughput, which can potentially be extrapolated to other nucleopiles moving forward. But one of the important aspects of this, right, is we wanted to dig or see how far this would dig into the cysteine ligandable proteome. So looking across multiple cell lines, we were able to see essentially nearly 30,000 quantifiable cysteine sites, again, using kind of the high accuracy quantitation of real-time search. We can then observe nearly 7.5% of all of the cysteines that we saw in a given system, in this case HCTs, were ligandable, right? So not every cysteine is ligandable, but a fair portion of them. And then of the total proteins, nearly 20% of quantified proteins observed in these systems had at least one ligandable cysteine. But all the same, it's still fantastic coverage of complex systems. And particularly what was striking about this is that even though many of these chemicals are small fragments, we often observed high degrees of specificity. So Millisecond showed very nicely and consistently throughout much of this data that you could find specific sites that reacted with just one ligand. So in this case, chlorocetamide 59 in the HCT 116 proteome. So there's 16,000 sites in there, and only one site essentially has a competition ratio that is above the threshold in terms of ligandability. So while there's thousands of cysteines profiled here, there's still also a large degree of specificity that Millisecond was able to observe in his data. And this went on, so you could look further at data within this. So P-loop ligandability in SRC kinase is one that Millisecond highlighted, right? So HCT 116, HEC 293s, and PA2s all showed essentially competition ratios for this chlorocetamide 126 on cysteine 280 in Src kinase that were highly specific for that protein, right? And so that was super encouraging because it's really consistent and it's really specific as a fragment. And so Millisecond then went on to show that while in essentially nanobret assays, right, we're looking for a competition of a reporter for a given either fragment or small molecule, that while the fragment alone, because it's very small, apparently could not displace the nanobret reporter, it could actively compete with a known Src kinase inhibitor, so SM71. So essentially, if you treat with the fragment alone, you don't see a significant shift, but if you treat with SM71, right, and the fragment, you see a reversal of SM71, essentially inhibition of reporter binding here, which then is indicative that chlorocetamide is seemingly binding within the same pocket as SM71, right? And so again, now we have a verified, validated, site-specific interaction of chlorocetamide 126 in Src kinase. And so essentially, what are next steps? Well, looking to the future, right? So as we start to use kind of these intelligent data acquisition strategies, one of the most important things for us is continuing to improve essentially the speed and rapidity with which we get through our data, right? So we want to do these large cohort studies, so more samples with more power. And we'd like to essentially also combine this with, be nice if the more samples we had, we didn't require larger and larger amounts of samples. So drilling down to smaller and smaller sample amounts, applying these to really small sample sets like single cells. We'd also like to essentially improve feasibility for applications of these intelligent data acquisition strategies in samples that have large analyte dynamic range, right? So particularly biofluids or biosamples, so plasma, et cetera. But even things like just looking particularly at reticulocytes or erythrocytes. We'd also like then to continue to kind of push the envelope on reducing instrument time. So if we can reduce and limit the burden of kind of expensive instruments, we can get through more samples and access more biology. And so with that, I'll just leave off. So I'll acknowledge folks in my lab, so Rose and Megan and Chris, who did much of the work from my lab that I talked about, as well as collaborations with Steve's lab. So I highlighted work from Milian Kulianen and Jiaming Li, as well as Jonathan Van Brinken. Collaborations with Jose, Cole, and Gary Churchill were also throughout. And a big shout out to Jimmy Yang, who we've worked extensively with to keep improving the real-time search platform, along with our collaborators at Thermo Scientific, who really gave us access to continue to build this development project. And with that, I'm happy to take questions.
Thanks, Devin. That was really fantastic stuff. Since I'm the moderator, I think I get first dibs on questions. So I have a ton here, but I'll limit it to just 2 for now, and I'll go in the order in which they were kind of made, a reverse order. So you talked about ligandability recently with cysteines in particular, and we just had Dan Nomura come and give a talk to our group last month. And one of the things that he kind of was the take-home point of his talk was that he thinks that every E3 ligase, or all the ones that he's encountered, are ligandable. And so he came up with this idea that virtually everything, and it came down to, in many scenarios, exactly the situation that you talked about, where there's one specific cysteine that is just particularly susceptible to this. And it seems to be that way. It's obviously very probe specific, but how much do you think is empirical, and how much do you think you can build off from things like AlphaFold to try and predict if there's ligandability in a structure-based kind of way?
Yeah, I think that's particularly interesting. We've had this debate a lot with basically like every ligand under the sun. You can think about like, oh, can we just use AlphaFold now to will that solve it? I think honestly, right now is kind of the field surrounding AlphaFold. The kind of brute force larger and larger fragment libraries are likely to be the faster way forward, right? So empirically defining those, but I think even with that, the ability to go in and structurally define essentially what domains or pockets might exist in these proteins is phenomenal. I think one issue that I've found, and you can see, I think I forget who now, I feel bad about that, but someone posted I think Chimerix Twitter account posted the top 144 or largest proteins in the human proteome structures predicted by AlphaFold, and a large majority of those have large unfolded patches that are kind of just random strings, right? Not random, but no really definitive structure there. So I think inherently there's still, because there's no full empirical knowledge of those, and because there's still kind of a knowledge gap in terms of what the actual structure is, the kind of brute force chemical approach is pretty viable now, especially because it's faster and easier. So
yeah, definitely. And Dan was showing that in many cases, in those big long strings that you see on AlphaFold, there would be one ligandable site that would be pretty important. And I'll ask one more and then handle it over. So this is kind of at the bones of real-time search. One of the things that you mentioned is when you do TMT and SPS, synchronous precursor selection, with an MS3, but there's smart fragment ion acquisition, do you still need to do SPS MS3? Like, if we know the ions, can we just selectively quantify on those ones and not even worry about co-isolating for the third step?
Yeah, I mean, so you could essentially, I think co-isolation is essential for us for sensitivity. That's the biggest thing that's still, but you could certainly say, if you want high specificity, right, just say, I just want the essentially fragment ion with the least contamination, essentially, of other peaks in the window, and I just want one. Mm-hmm. And inherently, even in SPS, right, we know that that'll give you the highest accuracy quantitation, but the lowest signal. And so that problem will kind of follow you with Orbiter real-time search, even in this context.
Very cool. All right, I'll turn it over and stop being so selfish now.
Yeah, nice talk, Devin. I have a couple of questions. So the first is related to TMT plexus. 18 is kind of what's out there, but is there a limit to the number? In other words, if you put more, isotopologues on the molecule, the mass itself increases, and then when the mass itself increases, if you try to extend the size of the reagent, is that the reason why there could be some limitation in the number of plexes, or is there any other chemical reason where synthesis of these isotopes, incorporating them, has become an issue or limits the number of plexes to 18? Is there some reason why it's only 18? Can't there be more than that? Can't there be 100 plex in the future?
So I think there can be more, but yeah, like you hinted at, right? It runs into the issue of the larger the adduct you put on there, the more you'll run into things like problems with chromatography and actually getting these super large, bulky adducts to elute off of LC columns. I think the current stack of TMT Pro is also kind of right, in part probably a legacy edition. So 126 through 131 was kind of where it was at, and so wanted to maintain kind of those pipelines, and it's also a nice size currently as it is. But yeah, clearly there's a limited number of carbons and nitrogens in there that you can use, so inherently there's a limit in the current set of molecules. But could there be another set? Yeah. Is the current kind of molecule or chemistry going to allow for that? Definitely not. And so maybe creative solutions. And I think, yeah, you also pointed to something too, where definitely the more complex the molecule, the harder it is to incorporate those stable isotopes at key positions.
But would the instrument have to be more sensitive in order to accommodate that kind of multiplexing as well, right?
Yeah, so I think even if it wasn't more sensitive, I mean, you can do kind of all the same things. Like the issue is you'll run into, I mean, if you only have one reporter charge that you're reading out per channel, like the ion statistics on that are just going to be terrible, right? So, I mean, that's essentially just a binary yay, nay, was this thing maybe present? So yeah, the more channels you add, the more sensitive you'll need to be, or you just need to be more conservative with kind of the intensity or the signal to noise that you use.
Okay. Yeah, I got some questions related to your talk. One was on the thermal stability profiling that you showed a volcano with the HDAC histone deacetylases that you identified. Mm-hmm. And so it's stability assay, right? So the expression, it's not the expression on the x-axis, it's the magnitude depends on the stability or instability. I noticed that it's a log fold of less than 0.5. Is that typical? And then another question tied to that is that there were only 3 HDACs. I don't remember what they were, 1.7, but like highly significant and had a magnitude above 0.5 that you use, right? So something, yeah, right there. Yeah. So, and then there are a whole bunch of other HDACs that did not change or right in the center of the ratio. So how do you look at the effects of affinity, target affinity with these inhibitors when some HDACs are active, some are not, or things like that?
Yeah, I think it's a great question.
And how do you determine based on a very subtle, difference?
Yeah, so the subtle differences or the small fold changes is addressing that is essentially to be able to confidently kind of define that. We're just trying to improve our stats, right? So improve our power for that, which is just more replicates, right? So that's part of why we'd like to use right, PSA. So we can do, in this case, if you did this with 2 conditions, right, you could do essentially 6 different replicates with the 18 plex, right? So that's just going to improve essentially our ability to confidently say whether or not a 50% fold change is meaningful or not. Second to that, yeah, so at least in part how we're beginning to rationalize this, and I think it's a large thing that we've talked with some other groups about too, is exactly what kind of ligandability and potentially inhibitory concentration or affinity, or exactly how those kind of interplay with protein stability, right? Thermal stability. Like what is the relationship there? And so we begin to explain it with things like this, where essentially most of basically the three that change all have a really low Ki, right? So that seems to be indicative that there's some specificity there. But I mean, if you kind of look in the under this, like an order of magnitude or two higher, and there's HDAC8, which essentially is not stabilized at all in these conditions. And similarly, HDAC3 is shown to have essentially the highest Ki and shows no effect here. So I think one thing that'll be important too is, especially for these highly regulated and potentially highly modified proteins, is trying to understand how their current state. So is there, say, a phosphorylation residue in here along the lines of kind of what the hotspot profiling or Judith Vian's work or Shelsevitsky's work has recently shown? Essentially, can we pull out kind of post-translational modifications and the effect of secondary regulation on protein stability and see if that's playing a fundamental role, and say, right, if there's in my mind, I go to kinases, but so phosphorylation event on the P loop of a kinase, does that inhibit some ATP competitive analog from getting to that, and so inherently doesn't have an effect on stabilization? So that's not really a factor of specific affinity for a given ligand, just a fact that the current regulatory state of that enzyme or protein in the current system, so lysate or cells, just is not amiable to a change in stability there. So I think there's a whole host of factors. This is more kind of the tip of the iceberg.
Yeah, but at the same time, I see the log P value. I don't know how you said the threshold of 0.05 or less, but it seems they're all close to the origin, right? So I don't know in terms of their confidence, whether they are all confident. I can't read it out from here. Is it above 1.3 or?
Oh, so like HDAC 3, 4, 7, and 8?
Yep.
Yeah, I'd say that I have no confidence that those are. I would say
so a negative log P value of 1.3 is about 0.05, right? So 95% confidence. So yeah, so even with the replicates that you ran, there's always some subproteome or some proteins that don't qualify. And do you think by having additional replicates that's going to improve this type of analysis, or is there something missing here?
So I think there's also a combination of things that we're particularly interested in, right? So one aspect of kind of doing the real-time search is the ability to kind of target things. And so if we have a rough idea of what we would like to go after, we could spend more time. In this case, you have an HDAC inhibitor. Can I just spend all of my high-res time, instead of spending it on kind of that gray portion here of proteins that don't seem to be significantly changing? Can I just spend it on proteins that might significantly change? So HDACs and then anything with a large fold change, and then spend way more time there trying to improve my ion stats so I can say confidently these proteins might be changing.
Yeah. And one final question. I guess others have questions. I have some more, but I'll just restrict to one more. Same with the competition ratio. Is how do you determine that? I mean, I saw that the one thing, that outlier that you saw was what you went after, and then you have some kind of cutoff that you marked in dotted lines in that plot. And I'm trying to understand like how you set those competition ratio limits. That particular protein stands out, but then there's also a whole bunch of others that are above the kind of the bulk of the proteins, right? That you identified.
Yeah, no, it's a good question. So I'll give you the punting answer, which is Ben Cravat decided and we followed suit. But also just the idea is essentially the competition ratio essentially scales to like about a 4-fold down reduction in kind of abundance of that as a
So
function.
those others might be off targets, right? So that might be problematic.
Totally, they could be off targets, and you can kind of assess it. So David Neusnow, who's a co-author on the paper, looked a lot at kind of skewness within this to try and assess specificity and off-target effects within this. And he's actually got some nice work. I don't actually have any figures for that in here, but to try and kind of build up for what's kind of the background distribution for these, and what might actually be indicative of kind of true off targets within that.
But am I right in this that there is no target?
So yeah, there's no known target. It's just a random fragment from a from a library. So
yeah,
very cool. Can you go back one more to the volcano plot? So one thing that I was always wondering, that this is the way that I always interpret it when we look at proteomics data like this in volcano plots, is that when you have 16 different channels, the y-axis suddenly becomes considerably more important than the x-axis. Is this incorrect? Like, now that we have so much confidence in the p-values, you no longer need to say that you need to be twofold over for it to be a significant change. When your p-value is an inverse log of 7.5, if you're 1 over or half over, that's a significant change, and you can be confident in those data. Am I interpreting that correctly?
I think largely. I mean, I think that's right now an open debate right now is like, do you still use the fold change cutoff? And I remember talking with folks back in postdoc.
What does Jonathan have to say about this? Because he gave me a hard time when we were collaborating at one time.
He's a strong-willed fellow too.
Yeah, there should be no random choices about what you do. You have to define it in, mathematically, right? So you can't put any thresholds on anything. It seems to be.
Did he do any power analysis on any of these fold changes to see what the threshold should be, and then the sample size and the effect?
Sample size was always 16. That was the optimal sample size. Or 18 now.
18 now. Okay.
Totally. I mean, yeah, we've done that. There's obviously issues because you would love to do one sample per 18, but of course you have almost no confidence.
Even in your other expression proteomics, I mean, it's not as bad as this, but you're looking at 0.5 fold changes. That means
so that's also inherently just a challenge of kind of these stabilization or PISA assays, is that all of it's a rare ligand you'll see that's gonna, I mean, it's actually impressive, right? You have a twofold change here with HDAC and nearly a twofold with HDAC2. That is pretty rare to see something that strongly kind of stabilized, right? If you go up and we look at kind of significance in these kinase inhibitors, right? Like the fold changes here are significantly lower, like much lower. Still significant, but much lower. And so there's a lot of, I mean, there's some factors here, right? Because you reduce technical variability by using the same lysate and then treating multiple different times across each one of the conditions or various things like that, right? So in this case, right, it's just literally the same lysate treated multiple different times and then added back in. So you can likely make an argument that you could go to lower fold changes, but exactly how low, up for debate. And actually, I think Jiaming and John are working on a nice set of work right now to try and define that, and say exactly how low you can go.
And if I could go to that last slide one more time with the KI, have you thought about doing it against KD? I mean, I know they're very similar, but KD seems to be like a more appropriate measurement that would be a better answer.
Yeah, this is a debate I was just having with Judith here, was can we just assemble kind of a huge list of KDs and look for kind of protein stability responses? And it's yes, but getting a large list of KDs is often not the simplest thing for every like protein you want out there. And then it's also right. So there's the kind of lighter purple HDACs that are here, but all those other proteins that seem to be off-target stabilization effects, and getting a KD or KI or anything for those is nearly impossible, right? Because no one's done that for some random off-target. So then how do you extrapolate for those? Where's the kind of relationship there? And so it becomes really challenging. So you can imagine then if you were to do, say, piece of the fragments where you have no idea in large part kind of what they're going to bind, how do you establish that kind of affinity to stability interaction? But I think it's a great question, and it's something, if you can solve that, please tell me, because I would love to see that. So. Problem solved. We can all go home.
Yep. I just have one small question.
It's good to open up the questions to the rest of the audience. I don't think we have too many, but I guess we would just ask them.
Yep. So obviously this isn't really too much my field, but you mentioned the Orbitrap by Thermo. Were there pros or cons to using that type of detector, or would a time of flight type of instrument be useful in this application? I was just kind of wondering what your mentality is. I mean, I know that Orbitraps can, increase your amount of sensitivity. So that's kind of what I was thinking.
Yeah, totally. So Orbitraps were predominantly used just to get their resolution, to get the full 16 or 18 plex, right? Because they're essentially neutron encoded for, there's like a 127N and 127C differentiation. But essentially I have a good picture of this on here, I don't think. But the TMT 16 and 18 will allow you to do kind of unit resolution to a pretty high degree. Yeah, it's not really a great picture for it.
So the isotopologues are like 6 millidaltons apart, right? So you had to have a resolution of close to 400 or above to resolve those, right?
But you can still ostensibly do the unit resolved ones, right? So the 127, the 126, 127, 128, 120, right? And then you could use a TOF or ion trap and get quant with sample multiplexing.
It's a great point, Steve. And along those lines, there is a big push, at least in industry, to start using the timsTOF, which is really, really fast. And it's the sort of thing where you can do a 2-for-1 deal in terms of the number of runs that you would get in the same amount of time when you're using high res to look at the 55K to separate these out, where they'll do 2 runs in 5 minutes each that will get 5,000 proteins or something like that. So I agree, Steve, that'd be really interesting to try these and skip a few of the isotopologues and see what kind of multiplexing you get.
Thank you.
All right, everyone. I think if there aren't any more questions, last call. No. If there are any more questions, let's.
I have a quick question on missing values. You say there's no missing values in TMT quant?
Fewer missing values.
Huh?
Fewer.
Okay. All right, that's fewer. Okay, it's fewer. Okay. We work with actually Jonathan on this missing value imputation, and he did this with label-free data, and he told me that there is no missing values in TMT, which I didn't quite understand. I was trying to get an answer from you.
Yeah, I think it's largely the idea is, given an intensity or like if you sum essentially the reporters in a given run to get a certain sum signal to noise, you should get essentially no missing values within a given plex. But you can run into some missing values, right, if you go between plexes. Right, you just didn't sample the same set of precursors.
Thank you.
All right, everyone. Well, let's virtually thank Devin for joining us.
Thank you, Devin. Nice
And
talk.
this was fantastic, Devin. Thank you so much. And we're going to keep an eye out for more real-time search applications in the future. Thanks a lot.
Thanks for having me.
All right. Bye-bye, everyone.
Bye.